I'd like to thank the ICMLF for the opportunity of speaking about mechanisms of disease progression in chronic myeloid leukemia. This was a talk that I gave in June 2022 for the ELN-EHA Specialized Working Group under the session Understanding the Pathogenesis of CML. Those are my affiliations. And let's jump right into this topic with definitions of accelerated and blast phase CML. These are the NCCN guideline criteria for blast phase and accelerated phase CML. And you can see that they're currently based entirely on clinical pathologic criteria. But what we want to focus on in this session is the mechanisms underlying the blast phase biology so that we can understand the contributions at the molecular level as to why we see these blast phase hallmarks in our patients: BCR-ABL independence, increased inflammation, increased stemness and differentiation block, and altered function of tumor suppressor genes and proto-oncogenes. And much of the data I'll present today was published by our group in Blood in June 2020 as a plenary paper. So you can refer to that for additional data if you should be interested. But what I'd like to highlight in this talk are three major learning goals. And the first is that blast crisis progression is characterized by the acquisition of a common or core transcriptome across the majority of patient samples. And this is true despite the significant heterogeneity at the molecular level we observe in blast crisis samples compared to chronic phase samples. The second is that the polycomb repressive complex, or PRC, and the processes that they drive contribute significantly to this core transcriptome. And the third is that a PRC-based model of blast phase is informative for the identification of chronic phase biomarkers and also potential therapeutic strategies. And both of these have obviously the possibility of direct clinical relevance. Now, the existence of a common or core transcriptome was first demonstrated by Jerry Radich in his seminal PNAS paper in 2006. And to understand the mechanisms driving progression, we asked two questions regarding the core transcriptome. How does this transcriptome contribute to the hallmarks I've mentioned in blast phase? And what genetic or epigenetic events induce this core transcriptome? These are two very important questions to try and address, because if we can do that, then we can suggest strategies to identify chronic phase patients at diagnosis who are at risk of poor outcomes, and perhaps even prevent or delay the emergence of blast phase and more effectively treat blast phase should it occur. Another important point I'd like to draw our attention to is that blast phase progression is characterized by significant genetic and epigenetic heterogeneity, and the existence of this common transcriptome strongly suggests the phenomenon of genetic or epigenetic convergence is occurring, and that identifying convergence points may lead us to better understand the mechanisms and therefore identify biomarkers and treatment approaches for the majority of patients with blast phase. Now, our approach was first to identify and decode the blast phase transcriptome and determine the expected and novel blast phase biological programs and how they arose from these transcriptome. And secondly, to perform an integrative multi-omics analysis to identify these convergent genetic and epigenetic pathways that are selected for during progression to blast phase. So what we did first was to define the blast crisis transcriptome using CD34 positive cells by performing microarrays on CD34 positive cells from chronic phase patients and also myeloid blast phase patient samples, and then used GSEA-based analysis, in this case using over 2,000 MSigDB gene sets to interrogate our transcriptome and see if they converge on the hallmark features of blast phase. Now here's a heat map of the statistically significant gene expression changes in our samples. This is chronic phase, this is myeloid, and this is lymphoid blast phase. And it is obvious at this resolution that there's a core blast phase gene expression signature, and that the myeloid and blast phase gene expression signatures are highly congruent, Most of the genes that go up can be seen in both transcriptomes, and a larger number of genes that go down again can be seen in both transcriptomes, with very few gene expression changes going in opposite directions between myeloid and lymphoid. And a third important thing I'd like you to draw your attention to is that the transcriptome appeared to be largely to transcend the mutations that we found in our blast crisis samples. The next question we asked was what hallmarks were encoded within this core transcriptome. And here, using GSEA and analyzing over 1,000 gene sets, we found, as expected, that leukemia stem cell signatures were enriched for in our blast crisis transcriptome, while progenitor signatures were depleted. And among novel hallmarks that we identified were inflammatory responses and cytokine signaling, and also relative quiescence. In addition, we also queried several hundred signaling, cancer, and epigenetic pathways to discover what was unique about blast phase. And one particular epigenetic gene set stood out. What we found was that blast phase cells gained PRC-related gene set signatures. Compared to chronic phase, both myeloid and lymphoid blast phase were enriched for the components of the PRC complexes. And importantly, and consistent with a gain of PRC activity, we found that established targets of PRC were enriched for silencing, since PRC silences gene expression. Now for PRC2, the opposite was true. We observed a loss of function type events with a decrease of the components of PRC2 and also a depletion for the silencing of PRC2 target genes. So here, let me take a slight detour and explain the function of PRC or the polycomb repressive complexes. A major function of the PRCs, of which there are two forms, 1 and 2, is to regulate stem cell activity and prevent differentiation. And they do this by enzymatically modifying histones and silencing genes via chromatin remodeling. Very often, human cancers are found to co-opt or hijack the PRCs so as to maintain a stem cell-like state in cancers. Now, turning to the blast phase genome, here we asked if genetic alterations in PRC components might be one mechanism which contribute to the PRC-related gene expression signatures we uncovered. And what we did, in addition to whole genome sequencing from samples from the same patients in chronic and blast crisis, was also exome sequencing. And we addressed this question by grouping genes affected by SNVs and copy number alterations into functional groups. So for example, in the PRC set of genes, we had 64 different genes comprising the genes encoding proteins that form those complexes. When we did this, we found that both for the whole genome and whole exome sequencing data sets, mutations affecting the genes in the PRC complexes were significantly enriched for, and when combined, the p-value is highly significant. What this was telling us was that there was selection for these genetic events for alterations in PRC1 and 2 activity. And here, I wanted to highlight the link between the genetic events we uncovered affecting the PRC components to well-known recurring chromosomal events in blast phase that had been documented in previous decades by our cytogenetic forebears. And the first thing we observed was that multiple PRC1 components seen here in this box were exclusively affected by copy number gains, that is, the red rectangles. Interestingly, many of these genes depicted by the ones with the red stars by them are also on the long arm of chromosome 17. This chromosome is frequently subjected to copy number gains in blast crisis in the form of an isochrome 17q, which is a major root ACA or additional cytogenetic aberration. And equally striking on the right-hand side, we see that the core component of PRC2, which is EZH2, is recurrently affected by both inactivating SNVs, depicted by the lightning rod, and deletions. And notably, EZH2 is situated on chromosome 7, which is frequently lost in blast crisis, particularly lymphoid blast crisis. More compelling, when we integrated our transcriptomic data, we could see that genetically and transcriptomically, PRC was exclusively affected by gain of function type events and increased transcription depicted by the dotted rectangles, and EZH2 by decreased gene expression. Altogether, our integrated results indicate the presence of strong selective pressure for gain and loss of function events affecting PRC1 and PRC2 respectively. We next turn to the blast phase DNA methylome and examine CD34 positive cells from chronic phase myeloid blast crisis and also normal bone marrow. And analogous to the core transcriptome, we identified a core blast crisis methylome seen in myeloid and lymphoid blast crisis and observed that this core methylome was largely indifferent to underlying genetic mutations. Next, we determined the function of the genes which are regulated by DNA methylation. And the way we did this was to demethylate blast crisis CD34 positive cells in vitro with decitabine, confirm their demethylation, and looked for the gene expression changes at those specific demethylated genes. Very surprisingly, only a small percentage of the demethylated genes were transcriptionally upregulated, as can be seen in these heat maps here. While relatively small, their functions were, however, important and included enhancing stemness and blocking differentiation. Importantly, what we also observed was that with TKI treatment, none of these changes were seen in terms of gene expression, indicating that these changes were BCR-ABL independent. We then asked, well, what other factors in addition to DNA methylation were regulating gene expression in blast crisis? Well, our combined genomic, transcriptomic, and DNA methylation analyses pointed strongly towards the PRC complexes. So we next asked what was the function of PRC1 and PRC2 in blast phase. And to assess this, we performed chip sequencing on 3 chronic phase and 3 myeloid blast crisis samples. Now, by performing chip-seq, we can actually determine the regions and genes that are bound by PRC1 and PRC2, and also if there's any overlap between other regions of interest, including regions that are heavily methylated. Immediately noticeable, even at this resolution, was that where EZH2 was binding across the genome here in chromosome 12, was that there appeared to be a strong association with DNA hypermethylated regions, an observation that we explored next. And what we did here was to integrate our methylome and chip-seq data, and we found that indeed EZH2 bound regions in CML occurred in an enriched form at hypermethylated regions, but not in hypomethylated regions. So what our results were indicating was that where EZH2 or PRC2 was binding in CML was instructive for DNA methylation in blast phase. This is a phenomenon that is termed epigenetic switching. So what we now think is that EZH2 binding in chronic phase CML over time and with unopposed BCR-ABL activity leads to DNA methylation and eventually the silencing of the genes at this region. We think this can be thought of as semi-permanent silencing and involves the differentiation and tumor suppressor gene activity. Importantly, once it occurs, cannot be reversed by agents that inhibit BCR-ABL activity. So this gives rise to our current model for blast crisis transformation and includes the following major features. From David Vitri's group, we know that EZH2 is induced by BCR-ABL activity and that it contributes to chronic phase leukemia stem cell reprogramming. Our model proposes that with prolonged EZH2 activity, EZH2 bound genes become DNA methylated, and are thereby silenced in a BCR-ABL independent manner. These include genes which are important in differentiation and tumor suppression. I did not show you the following data, but what I can tell you is that we also determined in the paper that increased BMI1 or PRC1 activity maintains silencing of a different set of genes, including novel tumor suppressor genes and genes involved in differentiation and apoptosis, also in a BCR-ABL independent manner. Now, based on this model, we tested 3 predictions. The first prediction was regarding novel drug combinations in blast phase. Here, we predicted that DNMT inhibitors combined with BMI1 inhibitors would be especially potent against blast phase. And what we found that as single agents, and particularly when combined, that they were able to significantly decrease the ability of blast phase cells from patients to form colonies while leaving normal bone marrow cells untouched. What we also found, using a seroreplating assay, the drug combination was able to particularly extinguish seroreplating, which is a hallmark feature of stemness. And a corollary we'd like to suggest is that because DNMT and BMI1 inhibitor combinations target the core transcriptome, we think that they may be effective in most patients regardless of underlying mutations that they may also carry. But of course, only clinical trials will tell us if this idea is indeed correct. Now, a second prediction we have is that BMI1 silences blast crisis tumor suppressor genes. And to identify novel tumor suppressor genes, we overlap genes which are silenced in the progression from chronic phase to blast phase, overlap this with genes bound by BMI1, and which were in turn de-repressed with BMI1 inhibitors. Now, taking a set of these genes, we found that the re-expression of one particular gene, the transcription factor NR4A2, resulted in the decrease of colony formation in CML cell lines, as well as primary blast crisis samples, and that this was dependent on its ability to bind DNA, since tumor suppressor activity was lost in a non-DNA binding mutant that we also obtained. Now, next, regarding potential biomarkers, we predicted that CD34 positive cells of chronic phase patients who are destined to transform to blast crisis will already begin to exhibit silencing of genes by DNA methylation and or PRC. Next, using gene expression data from 2 independent studies depicted here, we found that indeed chronic phase patients in these studies who did badly had enriched silencing of genes which were prone to PRC binding and DNA methylation in blast phase, those genes that were identified in our study. And to us, this suggested that pre-treatment patient samples in chronic phase could predict subsequent TKI responses in CML. Now, taking a slight detour, we also asked an important question. Do other mutations, including RUNX1, which is one of the commonest mutations to be observed in patients who undergo transformation, we asked the question, do these putative drivers also contribute to the transcriptome? Do they modulate other genes in addition to the core transcriptome? And I think the answer is probably yes. And this was described in the paper by Awad et al. From Dr. Satu Masjoki's group. They found that RUNX mutation containing blast crisis samples had a specific gene signature. And very importantly, they showed that RUNX mutation containing cell lines were sensitive to particular drugs and drug combinations. And using the same RUNX mutation signature I described, we were also able to corroborate their findings with our blast phase samples here. We were also able to refine the RUNX mutation gene set somewhat and found one that was also to be able to detect RUNX1 mutants, but to exclude lymphoid blast crisis wild-type RUNX1, which the other signature also tended to include. And as requested by the reviewer, in the paper that ultimately emerged from this result. We also found that our RUNX mutation signature performed relatively well in AML samples as well from the TCGA dataset. Here, the samples with mutations in RUNX, and here that are wild type. Okay, so finally, to summarize in terms of the concepts that we've uncovered, number one, blast phase progression is characterized by a common or core transcriptome that drives biologic behavior. We can see evidence of aberrant PRC1 and PRC2 activity, and also other drivers as well, including RUNX. We've determined that PRC-driven processes contribute to the core transcriptome. BMI1 and EZH2 regulate an overlapping but different set of genes that are involved in important features in blast phase, including differentiation block, stemness, apoptosis, tumor suppressor function, and both of these activities are TKI independent. We've also developed a PRC model of blast crisis progression that identifies putative biomarkers, prevention strategies, and combination treatments. Our findings also suggested that we could identify chronic patients who are destined to undergo blast phase transformation. So we tested this on a totally independent set of patient samples at chronic phase. Now, both the NCCN and ELN guidelines classify chronic phase TKI responses into three broad categories. Failure, depicted in the red line here. These are patients who ultimately will transform to blast phase. Warning patients who require switch in TKIs, or optimal responders who have very good responses to TKIs. Then we next asked if we could see blast crisis-like cells, or at least cells that had similar signatures to the ones identified in blast phase in chronic phase at the time of presentation. And looking at CD34 positive cells from the failure group and comparing them to the optimal group, we could indeed see similar signatures that we'd identified in blast phase, including the very blast crisis signature we documented, increased stemness, increased PRC1 components, increased inflammation in the form of gamma interferon signaling, and a decrease in proliferation or an increase in relative quiescence. So this result validated our integrated multi-omics analysis. And what we're doing next is using single cell sequencing and CyTOF to further examine these populations of cells from these different prognostic groups at the time of chronic phase presentation, to be able to identify other factors and better understand the biology that gives rise to TKI response heterogeneity. So I've come to the end of my talk here. And very importantly, I'd like to acknowledge a number of our collaborators. So these are the individuals in my group who helped drive the blast crisis story, particularly TK Ko and Lee Ken Leong. Our colleagues at the Genome Institute, led by Axel Hilmer and Asaf Javed. Very important clinical collaborators, Charles Chua and Tan Hien from the Singapore General Hospital, and also international collaborators, Chung-Wei Ju, Naoto Takahashi, Ravi Bhatia, and David Fruman. In addition, in the single cell work, we'd also like to acknowledge Vaidehi Krishnan from our group, additional clinical collaborators, as well as a team at the Genome Institute led by Sham Prabhakar. And in addition, ongoing collaborations with colleagues at SAMRI in Adelaide, Dr. Tim Hughes, Susan Branford, and Agnes Wall. Thank you very much.